And with that, I'm going to hand it over to Colleen and Kat. Thanks a bunch for coming, Colleen.
Great. Thank you so much, Abe, for the invite. I'm thrilled to be with you here today virtually. I only wish I could be there in person, but this opportunity has afforded me a chance to go through old photos of past trips to Acadia and take sort of a virtual field trip, if you might. So yeah, big shout out to our project partners on the Dragonfly Mercury project, including U.S. Geological Survey and the National Park Foundation, Appalachian Mountain Club, and Dartmouth College. Schoodic Institute has also been super integrally involved in this project since its inception. And I want to call them out as we're continuing work with them that I'll speak to at the end here today. So welcome, everybody. So this story actually starts with a story, and that story started 10 years ago on my first trip to Acadia as a NPS employee. It was 2010, and I had left Denver, where I'm based, for a month-long detail at the park to summarize the park's findings on Mercury. I had walked Cerdamont and saw my first Luna moth, stood in Stone Sound, witnessed Bill, who I know is on, Bill Golly, working his magic at McFarland Hill, 8 popovers at Jordan Pond, took a retreat at Schoodic, all the good stuff. And I guess you could say I've had a certain affinity for the park ever since. So half the joy in presenting today is that this project is very much entrenched and rooted in Acadia National Park. It grew out of efforts at Acadia. And furthermore, I got to take a walk down memory lane, so that was kind of fun. So several of the photos you'll see here are pulled from my personal files, and then others that have been shared by mainly Michael Marion, who has coordinated this effort at Acadia since. Also, and importantly, while at Acadia, I met a researcher from the University of Maine by the name of Dr. Sarah Nelson, who, through her work as a canon scholar and with the Schoodic Institute, have been working with school-age students in Maine and New Hampshire to crack the code on what species could serve as the proverbial canary in the coal mine for mercury risk in water bodies. While some sampling had occurred on park lands, the effort was more ad hoc. So the following year in 2011, Bill and I talked with Sarah at an Acadia workshop and suggested officially linking with the Park Service to start collecting at Acadia and a handful of other pilot parks later that season. And the effort has grown ever since. The Dragonfly Mercury project is a landscape scale surveillance study that links scientific efforts to understand the risks of mercury pollution with public engagement and management actions. The DMP, as you may hear me refer to it as, aims to increase the understanding of mercury contamination across the Park Service in the US, engage citizen scientists in the collection of dragonfly larvae, and inform Park Service policy and management decisions. We have since tiered off these goals, and I'll speak to that at the end of today's talk. But essentially, at the root, it's about engaging the public in actionable science through a combination of fieldwork and sophisticated laboratory analyses on the aim of linking mercury levels in dragonfly larvae to health risk for wildlife and humans, and in an effort to, at the pinnacle, manage and protect ecosystems. So this is an example of a place-based long-term monitoring network that has been catalyzed by USGS, centered on DOI NPS lands, a network that is critical for understanding a resource threat. And that threat is mercury. So due to its neurotoxicological effects, including altered behavior and reduced reproductive success, mercury challenges the very NPS mandate to preserve resources unimpaired for future generations. And if you're wondering, yes, this is a loon feeding dragonfly larvae to its chicks. I'm particularly thrilled about my finding this photo and being able to include it in this talk, because that is the link. Much of the mercury in remote ecosystems, such as national parks, arrives atmospherically after being emitted locally, regionally, or globally 5 coal-burning power plants and artisanal scale gold mines. These are 2 major sources of atmospheric mercury emissions. Medical incinerators and crematories also emit mercury and are human caused, but natural sources include volcanoes and other geothermal features. So we have emissions, transport, deposition, and effects. And we'll see mercury alongside other air pollutants come out in the form of rain, snow, fog, and dust. It's important to note, however, that this image doesn't show that inorganic or elemental mercury that is emitted to the air must be converted to the bioavailable methyl mercury on the landscape in order to accumulate in food webs. So this process of mercury methylation occurs by way of sulfate-reducing bacteria that essentially add a methyl group onto an elemental or inorganic mercury, thereby converting it to a bioavailable and toxic form, which is called methyl mercury. Not only does methyl mercury bioaccumulate, but it also biomagnifies, which means that it increases in concentration of the food web. This can be particularly concerning not only for wildlife, but also for, say, humans who consume fish and other top predators. Atmospheric mercury deposition is one of several potential pathways of entry of mercury to the watershed. Mercury deposition has been measured at Acadia at McFarland Hill, across from park headquarters, for over 20 years, thanks in large part to Bill Gawley and team. While there is a marked downward trend in the 3-year rolling average at Acadia, and we use the 3-year rolling average largely to account for the variability in precipitation. So we see the downward trend at Acadia over the past decade. Data indicate that the current rate of mercury deposition at Acadia is still about 2.5 to 10 times greater than what scientists think rates were before industrialization. So note that this figure and this downward trend particularly only account for wet deposition, that which comes out by rain and snow. So by excluding dry or fog deposition, however, total mercury deposition could be upwards to more like 20 micrograms per meter squared per year, which is 10 times the pre-industrial rate. That being said, given that mercury risk to the food web is decoupled from inputs such as deposition, while it is still an important metric to track, atmospheric mercury deposition alone cannot predict the risk of mercury, given that it must be converted to methyl mercury in order to be bioaccumulated and toxic to the food web. So dragonfly larvae provide a piece to the puzzle that serve as a beacon or warning signal of risk because they are predaceous. So they're eating a lot and accumulating mercury as they consume prey. They exhibit high site fidelity, which means they stay in one place more or less rather than, say, migrating like fish do. They're easy to sample, which is a really big deal for a bioindicator. And they are ubiquitous, so they are present on 6 to 7 continents. Moreover, interestingly also, they are present all over Acadia. So since 2009, 19 sites at the park have been sampled for dragonfly mercury levels for a total of nearly 1,700 dragonflies that have been analyzed for mercury from the park. This far surpasses any other park with the exception of possibly Olympic National Park. Acadia is also one of our longest running park participants, having contributed samples now for 10 years. So I'll be presenting some of the results from these sites today. What possibly sets the Dragonfly Mercury project aside from other place-based long-term research networks, however, is the engagement of citizen scientists and other community volunteers. Over 5,000 mainly youth participants have contributed well over 10,000 hours to this project since its inception. And the public is key to the project's success as they mobilize forces to collect samples from some of the most remote locations in the US. At Acadia specifically, close to 700 public participants have engaged in the study since we started tracking metrics in 2013. The years 2014 and 2015, as noted in these bar graphs, were particularly high points for project engagement when we had the funding to sample about 10 sites at Acadia 4 times each year under a joint USGS-NPS water quality partnership funding pot. And Ranger Michael Marion, here featured in the photo, took it upon himself to get citizen groups out on as many water bodies as he could during all 4 sampling periods, which theoretically is up to 40 water body events per year. One fall, according to Sarah Nelson, he managed to get every middle school on the island to bring a class out. Just super impressive. So talk about every kid in the park, huh? As we take a bit of a closer look at the types of citizens that have participated in the Dragonfly Mercury project at Acadia, this big chunk of the middle school groups explains the engagement of the middle schoolers in the project at Acadia, explains why we see a big chunk in the wheel here devoted to that middle school group. Most of the public participants are youth. We see not only middle school groups at Acadia, but also high school groups. I want to note that the patterns of youth engagement follow suit at the national level, which is exhibited on the right, except that at the national level, we see, on average, less engagement from middle schoolers and park programs and more engagement from high schoolers. But relatively speaking, still about 2/3 of the participants are youth. And thanks to Kat, we also looked at metrics about how many people join an outing and how many hours are contributed by each person at each outing. So on the left of the bar chart, you see Acadia in blue compared to the average by all parks in green on the right. And roughly we see about 5 people per outing, although I know that number has exceeded even 50 in some instances, which for the record is a bit too big of a group to sample, but can be done. But on average, more like 5, and each person generally is contributing about 2 hours per outing. We conduct post-sampling interviews with the parks to shed light on how the sampling season went. Some of the highlights, asking the question, what was the most surprising thing about the project? The level of engagement is one that rises to the top, about the ability of the project to not only provide a learning experience, but also to empower, to showcase enthusiasm and curiosity on behalf of the students and the kids participating, and to exhibit pride from student classrooms who receive the data back after they've collected in the field, completing the full research arc. Basically, get anyone down by a body of water with some nuts and waders, and everyone is bound to find something exciting, right? So at this point, I'm going to take a step back and just do a pulse check on the project's process and information flow pathways. If there's any immediate questions burning, happy to address those now, otherwise available afterwards for a more lengthy discussion. But yeah, we've been covering just up to this point, steps 1 through 3 on park engagement and the logistics and the sampling with public participants. But there's a lot that happens behind the scenes that gets us there. After a Park Service staff person is defined as the park lead or liaison, as we call them, for the project, we use a train-the-trainer model where we connect with that person who then connects with the citizen science group to collect samples. In the case of Acadia, that was mainly Michael. After the sampling effort, samples are shipped to the U.S. Geological Survey lab in Corvallis, Oregon, on dry ice for mercury analysis, where the samples are combusted while the dry ice is run through a direct mercury analyzer, or a DMA, for mercury analysis. So in step 4 here, you can see dragonflies prepared for combustion through that DMA. Then the data is analyzed. Step 5, interpreted and reported upon in data releases, data flyers, web maps, manuscripts, and presentations such as this one. This is step 6 in scientific outreach and communication. So that provides a ideal segue to discuss some of the scientific findings to date. One of the most important results from our landmark paper on this project that was published in Environmental Science and Technology this past summer shows that concentrations in dragonfly larvae are positively correlated with total mercury, or THG, as you see on both the Y and the X-axis, in 4 different fish gills. So you see fish mercury on the Y-axis, and you see the Eschnid equivalent total mercury on the X-axis, and that Eschnid equivalent is a way of which we normalize the concentrations of dragonfly larvae to a consistent family. So Eschnids are one of 6 families that we sample. This allows us, apples to apples comparison across sites and time periods. So we see in the different colors, each of the dots depicted represents an MPS site where both dragonflies and fish mercury data were collected. Blue are the piscivorous fish, green are the sunfishes, red are trout, and purple are the foragers, the prey fish. And these findings suggest that dragonfly larvae are effective indicators of mercury exposure in a range of freshwater taxa. Not shown here is the positive correlation also with 2 different types of amphibians, frogs and salamanders. But by looking at this, across these taxa and seeing these positive correlations across these taxa, it allows for the utilization of dragonfly larvae to provide robust comparisons of mercury bioavailability across a wide geographic range and among more habitat types than is typical with other taxa, like fish, for instance, that in ephemeral pools or ponds, there may not be fish populations, but we may likely find dragonflies. Thus, dragonflies provide another valuable tool for both comparing mercury concentrations among locations, as well as measuring response to perturbations and changes through time. Furthermore, we can use that relationship to estimate what's called an integrated wildlife and human health risk, an integrated impairment index, with regard to mercury across the Park Service and all units that have participated in the project to date. So depicted here is the site-specific risk in over 450 locations of 100 parks and protected places. What we see are in blue, the risk category that's below a known benchmark, and in green, we see a low risk category, and in yellow, moderate, and in red, high risk category, and in purple, dark purple, severe risk category. The pie chart on the lower left illustrates that while 10% of sites were below known impairment benchmarks for effects on wildlife and human health, about 12% of locations showed mercury levels that potentially exceed high or severe benchmarks for fish, wildlife, or human health, suggesting that fish in those sites may exceed EPA's fish consumption guidelines. So this risk is integrated, meaning that it considers these thresholds for both dietary intake of fish when utilizing dragonfly as a prey item and the risk to humans consuming those fish. And you can see by the map, the risk for mercury varies widely across the landscape. That's thought to be influenced by a combination of mercury emissions, deposition, localized influences like geothermal features even, landscape processes that control the entry of mercury into aquatic food web structure, and so on. Likewise, we can drill down and take a closer look at the impairment risk at Acadia. So mousing over now, we see the cumulative frequency of those risk categories. And then we can drill down closer at Acadia specifically. This is available, by the way, this USGS story map. And we can see that most sites at Acadia are either moderate or high, and there's even one at the severe risk category, which suggests that some fish species may exceed the EPA benchmark for protection of human health. So as evidenced by all the colors on the preceding maps, there is great variation in dragonfly mercury concentrations across the landscape. Again, here we see the same color scheme where green represents eastern, blue is central, and orange is west. And the bars on the right represent park mean for all parks that have participated to date, and the corresponding colors, green, blue, or orange. And the circles represent site means. So what we see is that total mercury concentrations, or THG, in dragonfly larvae vary widely both among park units as well as among sites within parks. In fact, we see concentrations from the highest site that are 135 times higher than those from the lowest site. And then likewise, within park, we see just as much variability. Again, we see that this risk may be a result of landscape factors and the inputs coming into the ecosystem. This extreme variation highlights the importance of site characteristics in determining dragonfly larvae mercury exposure, and it stresses the need for sampling across many sites to gain a better picture of relative mercury risk, facilitating the development of models to predict risk across unsampled water bodies in order to prioritize future conservation and research efforts, and even inform fish consumption guidelines, for instance. So drilling down again, we can look at the eastern US, which is essentially DOI regions 1 through 3, which includes the East and the Great Lakes, and assess spatial variation within that region particularly. We see here Acadia falling relatively within the top 10% of parks in the East and the Great Lakes as it pertains to the mercury concentrations evident at both park-wide and site level. But you also can note the variability within parks. So when you sample more in a park, as we have in Acadia, Acadia and Olympic are two core parks that have been established since 2014. We see more variation among the sites. The more you sample, the more variation we'll see. Likewise, another finding is that dragonfly larvae mercury concentrations also vary among the types of water bodies from which they were collected. So in red here, we see rivers, and in yellow streams. Wetlands are in green, and lakes are in blue, and ponds are in turquoise blue. And THG, or dragonfly mercury, is on the y-axis. So we see, interestingly, highest mercury concentrations from rivers and streams in dragonfly larvae than those from ponds and wetlands. I'm sorry, from ponds and lakes. On the bar chart just below, we're looking at the secondary habitat. So we can take a closer look at wetlands, for instance, and their influence on rivers or streams. So we see that while mercury in dragonfly sampled directly from wetlands was relatively low compared to rivers and streams, rivers, streams, and lakes and ponds surrounded by wetlands have higher dragonfly mercury concentrations than those without wetland influences. And this is often thought to be a result of dissolved organic carbon, which is a main driver of methylmercury production. So we can take a closer look at the map, which is on top. This is confounded a little bit. We see MDI, Acadia here. It's confounded a bit by the inclusion of Alaska and Hawaii. So sorry about that, they're on the left. But we can see that at Acadia, all primary water body habitats were sampled except for rivers. And so that's noted just by the corresponding color scheme. So yellow stream, green is wetland, turquoise blue, and blue are lakes and ponds. And that was intentional because Acadia was a core site along with Olympic, as mentioned. We were able to design the sampling to include a range of habitat types: streams, wetlands, lakes, and ponds. The only one missing is rivers. Now we can look a bit closer at the site-specific findings at Acadia. So the bars here represent the site average, and on the y-axis is total mercury concentration in dragonfly larvae, again, the Eschen equivalent. And the circles here actually indicate the family mean, so what we collected at each site. So as mentioned, there were 6 families that we sample. And interestingly, all 6 families have been sampled from Acadia at different sites. So what we see here is, likewise, as noted with the water body findings, that we see rivers with higher total mercury than lakes and ponds, and that same pattern is evident in the Acadia sites, where on the left, this is ordered from highest to lowest mercury concentration from left to right. Those above the park mean, and the park mean is denoted by that dashed line, are typically brooks and creeks, from Scudic Creek to Cromwell Brook and Richard Brook and Otter Creek, while we see more ponds and lakes below the park mean, such as Long Pond and Jordan Pond on the far right. So you'll note also amazingly high concentrations of dragonfly mercury at Scudic Creek, so much so that it goes beyond the extent of this chart. But this is based only on one sampling event in 2009, and it was only 4 individuals, and they were analyzed at a separate lab. So we are uncertain as to whether this is actually a good representation of mercury there, but it could be worth resampling. This was also, if you think back to that risk impairment map with all the colors, this was the severe impairment site, the one site at Acadia that was that dark purple. I'd also want to highlight real fast here Hodgson Pond, just below the park mean toward the right of the graph, and Marshall Brook 1, which is just above the park mean, mid-center of the graph, because it's those 2 sites where we have the longest continuous data record on dragonfly mercury at Acadia. So we have a 7-year record at Hodgson from 2011 to 2017 that's exhibited here in blue. And we have a 6-year record at Marshall Brook, 2014 through 2019, exhibited in orange. And so while our data records don't quite date back to this historic picture at Acadia, they establish a baseline and also allow us to track improvements or declines in mercury over time. Lake sediment cores, worth noting, however, do date back this far as that picture and even older. And at the end, I'll speak to a related study at Acadia looking at the history of mercury deposition both there and at Cape Cod. So it's important to continue this data record so we can track effectiveness. That is one of the goals, really, as we move forward with the Dragonfly Mercury project. There are sort of these 5 hooks that have been identified by way of a relatively newly established Dragonfly Mercury project steering committee, looking at risk quantification, so developing risk forecasting models to identify safer water bodies for fishing, informing management techniques and tools that could reduce mercury risk, assessing the effectiveness of domestic and international efforts to reduce mercury in the environment, so tracking improvements or declines over time, engaging and enhancing the learning of citizen scientists and the public, and communicating the importance and understanding of mercury issues for a variety of audiences. This is not just the steering committee evident here. This is part of the Science Dragonfly Mercury Project science team that met at Scudic in 2018. But this is a very pivotal moment, even though this isn't the entire steering committee. This is when it was fully decided. It was May of 2018. With Abe in the room and Michael and others, that we would press on and we would form the steering committee, and now we have, and it's been 2 years, and it's been rather successful. So while we have touted the relevancy of dragonfly data by our ability to feed into the resource condition assessment process, for example, providing direction to park management for each Park Service unit, we now aim to address in sort of this next phase of the project, the underlying so what questions. So namely, what can we do about it? Mercury is persistent, is revolatilized, re-emitted, it has a long residence time, it's not going anywhere per se. So how can we mitigate the problem? That being said, the application of the data to date, however, has been useful as it can inform parks regarding the severity of the threat of mercury. So due to elevated mercury values in each of these three pots on the left, deposition, potential for methylation, and risk to wildlife, Acadia falls in that severe, warranting significant concern category. Management implications include, the continuation of participation in this project, for instance, obvious continuation in the sampling of mercury deposition, monitoring for mercury in other park biota, and even communicating data on mercury and fish with stakeholders. So this project has created a generation of environmental stewards, enlightening a youth-based pool of citizen scientists about the connection of living things and the influence humans have on natural systems, and how smart decisions can protect our parks and the planet. But how do we grow up as a project? How do we achieve these larger objectives? So we as the committee and the leadership team on the Dragonfly Mercury project are exploring funding sources and partners. And frankly, expanding beyond MPS boundaries is certainly not out of the question, and really encouraged at this point. So part of the involvement of folks on the committee includes other agencies like Fish and Wildlife Service and EPA, for example, and we look forward to getting Forest Service more integrally involved as well. Getting existing data published will also bolster the DMP's aims, as in fact there are several Acadia-specific manuscripts in prep for publication, and I anticipate seeing the top two in journals sometime over the course of hopefully this winter or spring, and both draw on the core data from Acadia. First one is comparing Acadia with Olympic, and the second is looking at sort of a biota, so the water and sediment values, and correlating them with Dragonfly Mercury values. And it's also worth mentioning that Dave Evers from Biodiversity Research Institute and Celia Chen at Dartmouth and a few folks from the DMP team are writing up the synthesis of mercury data at Acadia. So this is long overdue, but we're excited to finally see some more progress on it. As mentioned earlier, Vivian Taylor from Dartmouth College will be publishing her work that assesses sources and history of mercury at Acadia and Cape Cod. Acadia also contributed to the Eastern Fish Mercury Study, which is a USGS-MPS joint effort. And while there have been some COVID-related delays in publishing this work, I'm hopeful that will be released in the spring as well. So, moving forward, we have work to do, which is good. Most importantly, we encourage participation. Certainly, Acadia is a super important unit to continue partnering with, given the long data record to date and the number of sites sampled, frankly, and the number of samples that have been submitted. As part of our plan forward, we are excited also to stoke our partnership with SCUDIC, and we will begin the planning phases for DMP-focused science communication workshops just after this meeting, in fact, which is exciting. So we're looking at ways to bolster our communication and outreach efforts and foster educational engagement. Of course, we need to write proposals and publish more papers, and definitely work the angles with partners to pitch this project at a broader scale. So here we are, continuing to grow and expand partnerships and work toward transitioning into that more established program. But of course, none of that would be achievable without the community built around this project. So it takes a village. Citizen scientists included, over 5,000 to date that have contributed to this effort. And of course, a very strong team, several of which are from Acadia, Abe and Michael and Bill even, and Kat, who's on the line, and again, our partners at USGS, Scudic and AMC. So I'm here representing all of them, but I thank you for your listening today and your attendance. And at this point, I welcome any questions or discussion. Abe, I'll let you take it away.
Yeah, well, thank you very much, Colleen. That was awesome. That was really clear and compelling. I mean, there's a tremendous amount of information there. I'd open it up to questions. You can feel free to put it in the chat or just unmute yourself. And I think we have a group here. I think you can unmute yourself and go ahead and ask. I'll wait a second let's see if anybody has a question, or I will start. All right. So one kind of starting basic question that I had is Mercury deposition. This is kind of going to the basics of Mercury deposition. Is it pretty uniform across the country, or is it kind of varied across the country, and at what scale is it kind of uniform? If it's coming from fossil fuel emissions, is it just falling everywhere, or is it concentrated in some places more than others?
Yeah, great question. So typically we've seen higher rates of deposition over time in the eastern U.S. as compared to the western U.S., and that's thought to be a result of the more localized and regional influences of, say, coal burning power utilities, like in the Ohio River Valley, for instance. However, that being said, we just see different signals. So we see more of that local and regional effect in the east. And then in the western US, we see more of a global contribution. So some of the newer work on Mercury is analyzing isotopes, which provide sort of a source fingerprinting. And we're able to better discern, global versus domestic. And we can see these signals coming that differ really from one half of the country to the next. Likewise, we see higher rates of deposition in mountaintops. So basically by way of, the orographic effect, you get higher precipitation at higher elevations. And so across the peaks of the Rockies and the Cascades and the Sierra, even the Smokies too, right? And the other mountain ranges in the east, you can see the influence of increased precipitation that likewise drives higher levels of mercury deposition at those elevations.
As if those high elevation species didn't have enough problems with climate change and other things.
Right.
Yeah.
Which
Ouch.
Likewise provides further impetus for utilizing those sensitive ecosystems as sort of a beacon for change.
Yeah. All right. Other questions?
Don't be shy. Michael, Bill, did I get it all right? What did I miss? Several pictures of Michael and one or two of Bill. So hopefully I interpreted that to the best of my knowledge. It's been a bit. Again, though, it was fun to sort of take a walk down memory lane.
I'll ask a question.
Yes, please.
So Mercury is really a really complex topic to communicate about to even other scientists, and I'm wondering if you can share any things you've learned about communicating about Mercury, and is that true that people, is it hard for people to get it, or are you finding that people get it, and what kind of ways of talking about Mercury have you found to be most successful?
Great question. I think that some people get it and some people don't. You know, I think it's just different, for instance, than other air pollutants that, say, my division that I work for, Air Resources Division, focuses on, like nitrogen, sulfur, whereby once deposited, you see or can observe an effect almost immediate. However, with mercury, there's that extra step that's essential. So it's decoupled, right, from deposition or other inputs by way of, effluent, for instance, from a facility. So that step is super integral to the process. And so it's confusing when one wants to look at, say, that mercury deposition map across the country and sees, higher deposition in the east and at mountaintops, but then sees that varied spatial pattern evidence that I showed with the Dragonfly Mercury concentrations that was kind of scattershot, like colors all over the place and overlapping sites. Even within one park, you go from severe impairment down to moderate and maybe even low risk, right? So more and more, we're looking to identify the drivers of these mercury values, because it's also not only about the inputs, but it's about the landscape characteristics, right? I find that visuals help. Sort of like, we're hopeful that by working with our outreach office, NROC, that we, in conjunction with them, will be able to create sort of a infographic that can be played that sort of shows how when, like the process, so the cycle from emissions to transport to deposition to effects, which, with effects, just include methylation. So it just includes those, or encompasses those more scientific-y, like jargony words like methylation and methylmercury. And so as much as we can simplify that, I think we'd be on par for better success in the outreach arena.
Yeah, I can speak to that too a little bit. I'm Kat, and I started out connected to this project because I was interning at Glen Canyon in Arizona, and then I slowly got drafted on to do more project coordinating level stuff. So I took out like Girl Scouts as like young as middle school kids, and sometimes even younger. And sometimes it's just like a fun outing experience for them, and they get to wade in water and find bugs, and that's kind of it. But you can kind of tell based on their questions that they're interested and they're curious and they're getting it. They always ask, like, where does the mercury come from, and what do dragonfly larvae eat and what eats them? And I think Sarah Nelson, she likes to say, she has a slide that says you are what you eat, and that's kind of a really good basic way of explaining it to younger kids. Yeah, we're always trying to kind of come up with more visuals, like Colleen said, and interpretive materials, both for us to communicate the hard mercury stuff, but then for all our park leads to have like flashcards about dragonflies and mercury and stuff like that to take them out of the field.
Thanks, Kat.
Another example of what we've done at Acadia has been to we put together an interactive program for the Nature Center that Colleen and I and a few other folks at both in the park and at Air Resource Division put together, and we've had that running for a couple of years, and that kind of goes through a few different pollutants, but mercury is one of them, and it's kind of divided into 3 sections with a segment on what is it, what's defining the problem, what does it do as far as the effects on humans and the environment and the ecosystem, and then how do we measure it, where we kind of scratch the surface about some of the monitoring and research that we do to gather that information. And I think that format be interesting to get some comments on what people actually took home from it. But from my experience talking to people that reviewed it, it's a pretty effective way of getting that across.
Yeah, that's good insight, Bill, because often there's that question of what can we do about it? Where does it come from, of course? And those questions alone sort of beg further inquiries into, well, if we have restrictions on coal burning power plants, and a lot of coal burning power plants in the country are being converted over to natural gas, we're seeing that now, shouldn't that be improving condition? And that underlies and underscores the importance of tracking that. So this is also part of, mention the tracking effectiveness. There's a global treaty on the Minamata Convention on Mercury. It's a legally binding treaty to assess how changes, in theory, reductions in mercury emissions are affecting mercury in the food web. So by obtaining sort of these baseline values in dragonflies that we can track over time, we can compare back and see, do we observe changes? So I think for, you know, I hate to say further research, but that would help, you know, solidify understanding of the connectedness between the inputs, like the sources and the receptors, really. But often, you know, what can you do? It's about conserving energy, right? You talk about what we can ask the public to do, or if they ask us, probably more likely, how can they affect the mercury cycle? You know, it used to be about batteries. Now there's no more mercury in batteries so much. But yeah, reducing our energy consumption largely in the US, although gold mines are the main source globally, but that's more so in third world countries.
Yeah, the what can we do about it is often the hardest question to answer, I find. And matter of fact, I was just referred a question from nps.gov from a young woman who was doing a project and specifically asked that about, you know, mercury issues in Acadia and, you know, some specifics and what can we do about it. And you know, that was the toughest part to answer.
Yeah.
And my response was basically, we have to keep up the monitoring and research and investigating the issue that we've been doing for the past 20 or 25 years, because all the information that we can gather on the sources and understand the problem a little bit will probably be the only way that we're going to solve this. It's a global problem that has to be solved, not like a local one that you can have somebody improve their septic system and the problem will go away. It's much bigger than that. And that's a tough thing for people to wrap their heads around.
Yeah, I mean, that's an interesting point, because we hope to, by way of continued work, to begin addressing sort of those mitigation options, because there are ways by which the landscape, not that we in the Park Service do a ton of management of our landscape per se. However, that being said, locally or even regionally developed mitigation measures that pertain to several influences in the Mercury methylation cycle. So wetland restoration, for instance. Wetlands can be a source of methylmercury. You know, prescribed burns, fires, wildland fire is also re-emits mercury that's been previously deposited, kind of mobilizes mercury in the soils. Even dams, which, you know, are more prevalent in the West, but the hydrologic fluctuations in reservoirs, consistent with releases of water over dams, induces the methylation cycle, because primarily where those sulfate reducing bacteria thrive. So it's well known that reservoirs have higher levels of mercury concentrations. So there's just a couple examples that we hope to better shed light on with collections of dragonflies over time and in those controlled settings, which may be well beyond Park Service lands too.
And that carries into some other questions that we have on other subjects when, you know, since we've had such crazy drought years lately, and we've had low water levels, and people ask about the effects of that and fluctuating water levels. And that's always something that I always try to inject into that conversation from that perspective as well too. So it's convoluted, but it's kind of cool.
Yeah, because what we see even, I didn't show this data, but you can find it on the story map, that, and I can put that slide back up here, the story map, our URL is listed here. But there's data that looks at the Dragonfly Mercury concentrations by ecoregion. And interestingly, so level 1 ecoregion, there's 10 of them throughout the conterminous US. And we see, and Alaska and Hawaii, but we see the highest levels of mercury in dragonfly larvae from the deserts, which is totally counter to what anyone maybe ever thought previous, because originally it was thought, oh, mercury is associated with water, and it is, but there's water in the desert. Yes, and what may be more evident in the North American deserts are these extreme flushing events, so these extreme wet dry cycles where you see like flash floods. When it rains, it really rains and it pours and it runs down a wash, and then it dries out almost relatively instantaneously, right? So that being said, it creates likewise to the rise and fall, or to the lowering due to drought, or to the lowering of a reservoir due to dam releases, this environment that's conducive to methylmercury production. So it's counter to prevailing thought.
Any other questions from folks? I think, Michael, it looked like at least at one point you were unmuted. Oh, there's Kevin. Kevin?
I don't have a question, but I just wanted to say, Colleen, great job in your presentation, and thank you for doing this. It's a great talk, and just really appreciate you making the time, and nice job.
Oh, thank you so much, and it's my pleasure. I'm honored to be here. Thank you. I just, again, wish I could be there, but I'll virtually be present.
Maybe this summer.
I know, maybe. Fingers crossed.
Any other questions folks have?
If you wouldn't mind, Mike, going a little bit off topic or off site, but what happened in Everglades? Because historically they had such a crazy problem with the extent that you were actually having mortality from mercury poisoning in panthers and things like that, and now they're to low and moderate levels. Is that a sampling artifact, or is something really happening?
Yeah, so I don't know if this is the best indication. That's a really good question. So I think it's totally site dependent. Some sites at even Everglades are lower than the others, which can be extremely high, right? And that, as I understand it, is completely contingent upon the presence of the canals, and really I think what drives the methylmercury production in that ecosystem is sulfate. So it's a sulfate driven system. What's thought to be, and evidence suggests, is from the agriculture by the surrounding Everglades Agricultural Association, I think it is, EAA. Anyway, and so it, again, just totally contingent upon location of that site. And I think what we're seeing is just an artifact of us not sampling the most sensitive sites, But great question, because alongside Acadia, really there's just been a ton of work down in South Florida on mercury and in panthers and in pythons and in gambusia and mosquito fish and now dragonflies, and it's very well studied.
And we were the first two parks, Everglades and Acadia were the first two parks to enter the Mercury Deposition network back in
right.
95 or 96 or whenever it was.
Right. And they see a lot of, Abe, you were asking about patterns, but I know we just have a minute left here, but we see a lot. I believe what's evidenced down in South Florida are by way of convective storms. You actually get a strong and high input of mercury deposition just because of the thunder cells that form. So they see elevated rates down in South Florida too of atmospheric deposition. And that's thought to be why. There's been some papers on it.
Yeah,
a good question.
Yes, very good question. And there are lots more that I think could be obviously asked. I think my understanding is this is the largest surveillance network of this type for mercury in terms of bioaccumulation of mercury in North America, and which is remarkable. And it's exactly the kind of, from our selfish perspective here at Acadia and with our research learning center and kind of emphasis of things here, this is exactly the kind of thing that we like to see and be a part of this, like piloting something and then watching it scale up. And you've done a great job leading that, Colleen, to something that's really nationally impactful. And so thank you very much for sharing it here with us today. And thank you everybody for attending. And I will circulate the, once I figure out how to post it. Well, feel free to email me if you want a link, if you want to be able to access the video. And I will also at least post it internally somewhere so that people can access it later. But thank you very much. Really appreciate it, Colleen and Kat.
You're very welcome. Thanks for having us. Have a good one, everyone.